Okay, so we are going to make a drawing now of something named the endomembrane system. Endomembrane, E-N-D-O, membrane, M-E-M-B-R-A-N-E, system. And the good thing about knowing the story of the endomembrane system is that this is what the vast majority of all of the organelles in the cell do. If you're a cell, your job, day to day, is in the most part how to make proteins, because proteins are enzymes, and enzymes then carry out reactions which make all the stuff that the cell might need. And so the vast majority of the organelles are going to be involved with making proteins. So if you know what the endomembrane system is, you can then describe what the vast majority of all the organelles in the cell do every day. And so the endomembrane system is going to begin with the nucleus. And the nucleus is the part of the cell where we're going to store the DNA. And so DNA, as you should know, contains all the directions for how to make proteins. And so our DNA is inside of the nucleus, and it's super important. It has really valuable information. It's so important that it never leaves the nucleus, and part of that is also because we only have one copy of all the information for the genes. So the DNA will never leave the nucleus. Instead, the information is passed onto something called RNA, and RNA is a really small little molecule, and we're going to have a whole 2 units where we talk about all the different aspects of this process. So for right now, just know that RNA is small, DNA is big, RNA goes out into the cytoplasm, DNA never leaves the nucleus. Okay, so the RNA goes out into the cell, and surrounding the nucleus is lots and lots of membrane. And this membrane is all folded upon itself to create a whole bunch of little cavities. And so one way to think about it is, imagine you have like a big bed sheet, and the bed sheet just came out of the dryer and it's all fluffy and warm and stuff, and you just lay the bed sheet on the ground. You just let it drop onto the ground. Well, when it drops onto the ground, it's going to leave all of these little pockets in between the layers of the bed sheet, and that's kind of what we're doing over here with this layer of membrane. This layer of membrane is called the Endoplasmic Reticulum. So endo, E-N-D-O, plasmic, P-L-A-S-M-I-C, reticulum, R-E-T-I-C-U-L-U-M. So endoplasmic reticulum. And the endoplasmic reticulum is where proteins are going to be made. And they're made from the information that is inside of this RNA. And another little, it's kind of like a protein, kind of like an organelle. It's very small, but it's called a ribosome. And ribosomes are going to take that information from the RNA and turn it into a protein. And again, that's a whole process. That's like a whole test over that whole thing. But we have this area of the endoplasmic reticulum where you've got nothing but a whole bunch of ribosomes attached to the ER, and they are cranking out proteins. And so here's our ribosomes, our ER. And because you've got some areas where you find the ribosomes and other areas where you do not, we've got 2 different like zones within the ER. The area where you find all of the ribosomes, this area, this is called the rough ER, because it would look rough in appearance. It's going to look like it has a whole bunch of little bumps or zits on it. And then down here where you do not have the ribosomes, this is known as the smooth ER. Okay, so the way that the endoplasmic reticulum is going to work is it's like, here's your membrane of the ER. Your ribosome is going to dock onto that membrane, and it's going to dock on that membrane with the RNA attached. The ribosome is then going to turn that RNA into a protein. So we've got our protein being formed down here. But the protein is going to be placed within that fold in the ER membrane. And so these open areas of the membrane we call the lumen. And lumen is a really generic word. Lumen just means empty space. And so we could say lumen of the mitochondria, or lumen of the nucleus, lumen of the cell itself, whatever. Lumen just means empty space inside of. And so in this case, we've got the ER, It has a lumen and empty area. The ribosome is going to turn the information from the RNA into a protein, and that protein will be deposited within the lumen of the CR. The protein is then going to fold. It's going to fold in a way that it's able to predict because of the R group attractions within those amino acid chains. And you're going to get that mature, folded tertiary structure. But because that folding is really complex and takes time, that's why it needs to have this nice quiet space where it can occur, which is going to be this lumen of the ER. So the endoplasmic reticulum is where proteins are going to become mature. It's where they're going to become functional. Next, as they become mature and functional, the membrane is going to bud out, so it's going to accumulate in little areas of the ER on the outside. Then the membrane will just bulge out and pinch off and go into the next part, which is called the Golgi Apparatus. And the Golgi Apparatus is still just membrane. It just looks a lot prettier, a lot more organized membrane. And so this right here, this is the Golgi Apparatus. And the ER had the smooth and the rough areas. The Golgi has got an incoming and an outgoing side. So the incoming side is called the cis face, C-I-S, cis. The outgoing side is called the trans side, T-R-A-N-S. So the Golgi apparatus has a cis face and a trans face. Cis is incoming, trans is outgoing. And what's happening is that as the products of the ER arrive at the Golgi, they initially are just all dispersed all over the place. You got all these proteins, they're all mature, they're all ready to go, but they all have completely different functions, they all have completely different shapes, they all work for all different sorts of things. Maybe one goes to the mitochondria, maybe one goes to the membrane, maybe one is supposed to stay within the cytosol and float around and do stuff. So they all have these different destinations. And so what's going to happen is they're going to start to clump themselves, not according to type necessarily, but according to their destination. Where is it that they want to go? And you're going to end up with these concentrations of proteins in really particular spots to where all the proteins maybe in this area are going to go to the cytoplasm. Maybe all the proteins over in this area, they're on their way to the mitochondria. And then as they get into those particular locations, the membrane will start to pinch in. It'll kind of bud off, and you're going to create this little packet of membrane that has proteins inside of it. And all the proteins inside of that little piece of membrane, they all need to go to the same spot. Well, these little packets of protein, or of membrane, those are called vesicles. V-E-S-I-C-L-E-S, vesicles. And those vesicles now need to go somewhere. Well, vesicles by themselves can't move. They don't have like little rocket packs or anything to move around the cell. So they need something to take them from one place to the next. And so they need to get on a little railroad. And the cell has little railroad lines that go from the Golgi to all throughout the rest of the cell. And it might be over to another organelle, maybe it's to the outside of the cell, whatever. And these little railroad lines, these are part of the cytoskeleton. And the ones that are going to be involved in this process are called microtubules. M-I-C-R-O-T-U-B-U-L-E-S, microtubules. And microtubules are chains of protein that are going to just stretch out randomly. And when they shoot out into the cell, if they don't touch anything, they just retreat. They come back to where they originated from. But instead, if they actually hit something, then they're going to become stabilized. And now they're going to be fixed. And now you have a point of contact from the trans side of the Golgi to whatever organelle might be out there, the vacuole or a lysosome or whatever it might be. So these vesicles, as they get near a microtubule, they're going to become attached to it with another little protein, which is called a motor protein. And so in this picture, I got my vesicle, and then my motor protein is just these, it's like a Y that has two little circles on the end, and it's upside down. And the motor protein, right here, I'll show you guys videos in class and everything like that, but the motor protein kind of is like this little guy with huge clown shoes, and because of instabilities or stabilities within the protein, it's going to move those little shoes to kind of walk along the microtubule. And as it walks along, it's going to be carrying this vesicle behind it. And so that's going to end up delivering whatever contents of this vesicle to the appropriate organelle. And so if we kind of look at what's going on, we've got the endomembrane system. Endomembrane because it's within the cell, endo, and then membrane because really all that's happening here is you're moving membrane from one place to the next. We start with kind of like the research and design section, the nucleus that has the DNA inside, a message or a blueprint of what they want to make is put into the RNA. It is then processed on the factory floor, which is the endoplasmic reticulum, where you're going to make that protein. It's going to become mature. And now that package, that mature product, the protein, which folded within the rough ER, is going to go over to the Golgi apparatus. The Golgi apparatus is kind of like the warehouse of a factory. You've got all these products, now you need to put them into boxes, which are the little vesicles, and get them all sorted into the locations they need to go. Once they get sorted into the little boxes or the little vesicles, now the little delivery trucks, the motor proteins, are going to deliver them out into the cell wherever it is they need to go. And so we're going from the from the nucleus to the ER to the Golgi to the vesicles, from left to right. And this is how we're going to be able to have this endomembrane system.